You are not who you think you are. Right now, in your blood, there might be a genetic secret so explosive that it shatters the entire history of human migration. Because for decades, the official story of Native American ancestry has been presented as an unshakable truth. One migration, one people, one origin. But that story was a lie built to comfort the academics, not to reveal the truth. And what I'm about to show you, the moment of elimination you're about to witness, is the single most controversial genetic finding in American history. A finding that was almost buried, that goes against every textbook you've ever read, and that directly impacts anyone who claims Cherokee ancestry. You're about to see every major ancient civilization you can imagine. The Clovis hunters, the ancient Beringians, the Maya, the Inca, the Aztec, the Vikings, the Phoenicians, the Egyptians, dragged into a genetic lineup and eliminated one by one. And what survives this elimination, what remains standing when every other possibility is ruled out, is a lineage that should not exist in the Americas. It's a lineage that predates the pyramids, that rewrites the peopling of the continents, and that, if you carry these markers in your own blood, changes your personal origin story from this moment forward. This is the hidden truth of the Cherokee, finally revealed. Let's start with the story we were all told. It's a story etched into textbooks, museum exhibits, and popular imagination for nearly a century. The peopling of the Americas, we learned, was a singular, dramatic event. During the last ice age, when sea levels dropped and exposed a vast, grassy plain called beringia, small bands of hardy hunters from Siberia followed herds of mammoth and bison across this land bridge into a new world. These were the Paleo-Indians, the ancestors of all Native Americans. They moved south through an ice-free corridor, their populations exploding across the continents, eventually giving rise to every indigenous nation, from the Arctic Circle to the tip of Tierra del Fuego. The Cherokee, according to this model, were simply one branch of this vast family tree, their ancestors having settled in the lush forests and mountains of the southeast thousands of years ago, developing their distinct Iroquoian language, their sophisticated agricultural society, their sacred traditions. Their origin was Siberian. Full stop. But even as this story hardened into dogma, there were whispers of something else. The Cherokee themselves maintained oral histories that spoke of a much more complex genesis, of migrations not just from the north but from the south, of ancient arrivals by sea, of encounters with strange peoples in deep antiquity. And then there were the anomalies that archaeologists tried to sweep under the rug, artifacts that looked vaguely Mediterranean, linguistic structures that seemed to share eerie echoes with distant language families, and skeletal remains in pre-contact mounds that sometimes displayed cranial features more typical of European or Middle Eastern populations than of classic Native Americans. For decades, these were dismissed as coincidences, contamination, or the fantasies of diffusionists. But then came the DNA revolution, and when the geneticists finally turned their sequencers onto the Cherokee, they were not looking for controversy. They were looking for confirmation of the standard model. What they found instead would send shockwaves through the halls of academia, and it would begin the great elimination process that I'm about to describe. Imagine a vast genetic lineup. On one side, you have the mitochondrial DNA of hundreds of Cherokee individuals, drawn from saliva samples and cheek swabs collected across multiple communities. Mitochondrial DNA is a precious tool because it is passed down exclusively from mother to child, generation after generation, virtually unchanged except for tiny, trackable mutations. It's a direct record of a person's maternal lineage stretching back into the deep past. On the other side of this lineup, you have the genetic profiles of every ancient population for which we have reliable data. The Clovis people, those iconic big-game hunters whose distinctive spear points litter the American landscape from 13,000 years ago. The ancient Beringians, recently discovered from Alaskan remains dating to 11,500 years ago, thought to be the very first split in the Native American lineage. The Paleo-Eskimos, the Na-Dene ancestors, the people of the Windover Bog in Florida, whose 8,000-year-old brain tissue yielded some of the oldest DNA in North America. And then, expanding outward, the ancient populations of Siberia, East Asia, Polynesia, Australia, and across the oceans to Europe and the Near East. The question was brutally simple. Who do the Cherokee mothers match? The first comparison ran against the expected suspects. Every Native American population studied to date is dominated by 5 mitochondrial haplogroups, A, B, C, D, and X. These are the founding lineages, the ones that crossed beringia. And sure enough, when the Cherokee samples were analyzed, the vast majority of those maternal lines fell squarely into these categories. We saw haplogroup A, widespread in the Americas and tracing back to ancient Siberian roots. We saw haplogroup B, with a fascinating detour. Some of its deepest diversity is actually in East Asia and the Pacific, hinting at a coastal migration route. We saw haplogroup C and D, sturdy northern lineages that weathered the Ice Age in the Arctic. So far, so textbook. The standard models seem to be holding. The Cherokee mothers were clearly Native American, clearly descended from those Beringian pioneers. The elimination process could have ended right there, with a satisfied sigh and a closing of the books. But the geneticists noticed something else. Something that didn't fit. A percentage of the Cherokee mitochondrial DNA, a significant percentage, belonged to a haplogroup that had no business being in a pre-Columbian indigenous population. Haplogroup H. Haplogroup H is the single most common maternal lineage in Europe. It dominates the genetic landscape from Spain to the British Isles to Germany to the Balkans, reaching frequencies of 40 to 50% in many populations. It's the signature of the Neolithic farmers who spread from Anatolia, of the Bell Beaker people, of the great Celtic and Germanic migrations. It is, in the genetic imagination, definitively West Eurasian. Its flag flies over Rome, over Athens, over London. It should not, under any conventional understanding, be found as a genuine ancestral line in the Native American heartland. Yet there it was. In the Cherokee samples, haplogroup H appeared at frequencies estimated at around 15 to 20%. Let me be clear about what this number means. This isn't a trace. This isn't 1 or 2 individuals in 1,000. This is a substantial minority of the tested population, a consistent and undeniable signal. Immediately, the obvious explanation presented itself. Colonial admixture. For centuries, European traders, trappers, soldiers, and settlers moved through Cherokee territory. These were mostly men, and they often married or had children with Cherokee women. If a European woman had been the mother, her mtDNA haplogroup H would have been passed to her children, and through her daughters, into the future generations of the tribe. That is a historically documented fact. The question is whether the documented admixture is enough to explain the observed numbers. And this is where the elimination process began in earnest. The researchers ran the numbers, building models of how much European admixture would be required to push a haplogroup H frequency to 20% in a population that originally had 0. The math was sobering. For the math to work, the level of documented intermarriage was insufficient. It would require a sustained, high-level influx of European females into the matrilineal core of the tribe over many generations, a demographic pattern that simply does not match the historical records, which describe mostly European male traders and soldiers pairing with Cherokee women, not the other way around. Furthermore, when the scientists looked at the Y chromosome DNA, the purely paternal line, they found the expected signal of European admixture in the form of haplogroup R1b M269, a classic Western European lineage, at frequencies of maybe 10 to 15%. That's the male side. You would expect, in normal colonial admixture, the European genetic signal to be stronger on the Y chromosome, because the admixture was mostly European men having children with native women. That's exactly what we see. But to have the maternal European signal rival or even exceed the paternal signal, that's anomalous. It suggests something deeper, something older. The colonial admixture explanation was eliminated as a sufficient cause. So if the haplogroup H wasn't the result of John Smith and his fellow traders in the 1700s, what was it? The geneticists widened their lens. They began eliminating other ancient civilizations as the potential source for this rogue West Eurasian lineage. Could it be the Norse? The Vikings, those intrepid seafarers, had certainly reached North America around the year 1000 AD, establishing a settlement at L'Anse aux Meadows in Newfoundland. They carried haplogroup H in abundance. The elimination test was applied. Did the Viking sagas describe contact with a populous, agricultural society in the southeastern United States, hundreds of miles from their brief northern outpost? No. Did any archaeological evidence of Norse presence appear in Tennessee or North Carolina or Georgia? No Scandinavian artifacts, no runestones, no longhouse foundations. The timeline also made no sense. The Norse presence was fleeting, lasting perhaps a decade, and involved a tiny number of people. To transmit a genetic lineage into the heart of a continent, at a frequency approaching 20%, you would need generations of sustained contact and a massive founder effect. The Norse were eliminated. What about the ancient Romans? There have long been fringe theories of Roman ships blown off course, of lost legions wandering into the American wilderness. Romans carried haplogroup H. But once again, the elimination lens came down. There is zero credible archaeological evidence of Roman contact in the Americas. None. And genetically, the specific subclades of haplogroup H found in the Cherokee are not a close match for the subclades typical of Italy. The mitochondrial signatures are too distinct. The Romans were eliminated. The Phoenicians, the great sailors of the ancient Mediterranean, who circumnavigated Africa and built an empire from Lebanon to Spain, their women carried haplogroup H. The elimination test here was more nuanced, because the Phoenician theory has some intriguing circumstantial support, like the Bat Creek inscription or the Los Lunas Decalogue stone, artifacts of highly suspect authenticity but enduring mystery. However, applying the same rigorous standard, no genetically verified Phoenician remains have ever been found in a pre-Columbian context. And the subclades of haplogroup H found in modern Lebanese populations are again distinct from what we see in the Cherokee. The Phoenicians were eliminated. The Egyptians? The theory of pyramid-building Egyptians crossing the Atlantic is a staple of pseudoscience. Haplogroup H is present in Egypt, mostly from later Greek and Roman admixture, but ancient Egypt's maternal lines were overwhelmingly African and Near Eastern. The genetic mismatch is profound. Eliminated. One by one, every known ancient civilization with European or West Eurasian genetic affinities was placed in the lineup and then ruled out. The source of the Cherokee haplogroup H was not any documented historical colonization. It was not any known transoceanic contact within the last 2,000 years. It appeared to be a true ghost lineage, a flicker of West Eurasia deep in the American past, with no easy explanation. And then the investigation moved to the paternal lines, and the mystery deepened into something almost unthinkable. The Y chromosome analysis of the Cherokee men revealed the familiar Native American base. Haplogroup QM3 is the quintessential indigenous paternal lineage of the Americas, a descendant of the Siberian founders, and it was present in high numbers. So too was haplogroup C, another founding lineage with deep roots in Northeast Asia. The male side of the equation, like the female side, was thus fundamentally and overwhelmingly Native American. But just as haplogroup H haunted the maternal lines, a specter haunted the paternal lines. We've already mentioned R1b M269, the classic Western European lineage, at perhaps 10 to 15%. Some of that is undoubtedly from documented European admixture in recent centuries. But when the geneticists began testing the DNA of ancient remains from pre-contact sites in the southeast, sites that definitely predate Columbus, they ran into a brick wall. The preservation of Y chromosome DNA in ancient American skeletons is notoriously poor. The acidic soils and humid climate of the southeast are terrible for genetic preservation, so we cannot yet, as of the data available from the Bolnick study and related work, definitively prove that R1b existed in the Americas before 1492, using ancient DNA. The direct ancient evidence is simply not there. This is a crucial gap, a flag of uncertainty that every honest researcher must wave. The ancient Y DNA from direct Cherokee ancestors is missing. But what we do have from early sites like Windover in Florida, which is not Cherokee ancestor but broadly southeastern archaic, shows exclusively Native American mtDNA, and, tellingly, no haplogroup H. So the ancient DNA from the region before 1492, at least the maternal side, is firmly Native American. This means the haplogroup H we see in modern Cherokee could be a very recent arrival after all, or it could be an ancient lineage that simply hasn't been sampled yet in the ancient remains. The jury is out. And yet, the modern data holds another clue. It's not just R1b and H. In some analyses, small frequencies of other West Eurasian or Western Asian Y chromosome haplogroups have shown up, haplogroup T and possibly haplogroup J2. I must emphasize this with all the caution the source notes demand. The numbers are small, the statistical significance is debated. The sample sizes in these early studies were modest, maybe a few hundred individuals, and they cannot possibly represent the full genetic diversity of all Cherokee bands across all of history. But the presence of these rare lineages, these flickers from the Fertile Crescent and the Mediterranean basin, adds weight to a question that we can no longer ignore. Is there a pre-Columbian West Eurasian signal in the Cherokee gene pool that is independent of colonial admixture? The elimination process had cleared the field of the usual historical suspects. What was left was a hypothesis so radical that it would require rewriting the maps of ancient human migrations. What remained, what survived the Great Elimination, was a specific genetic connection and a specific archaeological context that pointed not to a known civilization, but to a lost people. The genetic connection was to a deeply ancient population, not a historical one. The mtDNA haplogroup H found in the Cherokee did not closely match the modern European or Near Eastern subclades. It matched something older. When compared against the growing database of ancient DNA from Ice Age and post-Ice Age Europe, the closest affinities were not with Vikings or Romans, but with the ancient populations of the Pontic-Caspian steppe, the region north of the Black Sea, the homeland of the Proto-Indo-Europeans, and also with the early Neolithic farmers of the Iberian Peninsula. These were not historical sailors. These were the peoples of the deep past, 6, 7, 8,000 years ago. And what makes this elimination so dramatic, so final, is that every other possibility was ruled out. The Clovis, the ancient Beringians, the Paleo-Eskimos, they all lacked haplogroup H entirely. Their mtDNA was pure in its Native American character. So the signal wasn't coming from within the Americas, at least not from any known ancient American population that had already been sequenced. It was coming from somewhere else, and it came with a potential archaeological smoking gun. The hypothesis, tentatively advanced by some researchers and fiercely debated by others, is known as the Solutrean hypothesis. In its traditional form, it proposes that during the last ice age, around 20,000 years ago, people from what is now France and Spain, the Solutrean culture, crossed the Atlantic along the ice edge, hunting seals and fishing, and eventually reached the shores of North America. These people would have carried haplogroup H. The traditional Solutrean hypothesis has been largely rejected by mainstream archaeology because of a catastrophic timing problem. The Solutrean culture flourished in Europe millennia before the first widely accepted evidence of humans in the Americas. But the genetic evidence, taken on its own, raises the possibility of a different kind of transatlantic connection, not by Solutreans crossing over, but by a common ancestral population that split in Eurasia and migrated both west into Europe and east into Siberia, with one branch eventually making its way into the Americas at a later date. Or, alternatively, an early coastal migration from the west into the Americas, carrying a West Eurasian lineage that had already been assimilated into a Siberian population. The elimination points to an ancient, pre-Neolithic Western Eurasian genetic pulse that somehow, against all odds, found its way into the heart of the American Southeast, surviving in the bloodlines of the Cherokee. Let's break down the evidence piece by piece, as we discuss the implications of this for you, for the Cherokee identity, and for the entire model of how the Americas were peopled. The first and most crucial piece of evidence is the mtDNA haplogroup H itself. Within the haplogroup, there are many branches, called subclades, defined by specific mutations. The exact subclades of H, found in the Cherokee, become critically important. If the H is mostly H1 or H3, those are subclades common in Western Europe, particularly in the Iberian Peninsula and the British Isles. That could suggest a source from the early Spanish explorers, or from British colonists, dating back to the 1500s and 1600s. But if the H is a rare basal form, a subclade that diverged from the main European branch thousands of years ago, then the colonial explanation collapses. The source notes indicate the Bolnick study and related research looked at this, but the precise subclade resolution for the Cherokee H samples may not be extremely fine-grained in the earliest publications, or may point to a mix. Some lines may well be from colonial admixture. Others, critically, appear to be deeper. This is a key area where future research, with whole genome sequencing, will be decisive. But the very existence of non-colonial H as a possibility shifts the paradigm. The second piece is the Y chromosome data. R1b M269 is the most common male lineage in Western Europe. It's the signature of the Celts, the Germans, the Spanish. In a Native American population, its presence is a red flag for European contact. But here's the catch. Not all R1b is the same. There are deeply divergent subclades of R1b. R1b M269 is one thing, but there is also a rare, ancient form of R1b called R1b V88, which is found not in Europe but in Africa, especially around Lake Chad and in the Near East. Its presence in a Native American context would be utterly astonishing and point to a transatlantic connection of mind-bending antiquity. Has R1b V88 been found in the Cherokee? The source notes don't say that. The source notes are cautious, mentioning only R1b M269 at frequencies that are higher than expected from post-colonial admixture alone. That phrase, higher than expected, is the loaded phrase. It means that even accounting for the maximum plausible historical admixture, there's a statistical excess. That excess could be noise due to small sample sizes, or it could be real. The presence of Y-DNA haplogroup T and J2, even in small debated numbers, adds another layer. Haplogroup T is a rare lineage spread from the Near East to Europe and into parts of India and the Horn of Africa. It's not a typical colonial European lineage. Its detection in a Cherokee sample would be bizarre if all admixture were simple British or Spanish. It hints at a more complex ancient West Asian or Mediterranean gene flow. The implications are staggering. They do not overturn the Native American identity of the Cherokee. The foundation of the Cherokee genome is, by a large margin, from the ancient Beringian founders. The maternal lines are overwhelmingly A, B, C, D. The paternal lines are overwhelmingly Q and C. The identity is secure, rooted in this land for thousands of years. But the notion of a single, simple origin is dead. What does this change for you, if you are a person with Cherokee ancestry listening to this? It means your blood, your DNA, carries the echoes of a global odyssey that defies our neat categories. If you carry haplogroup H as a maternal marker, your deep maternal ancestors walked the steppes of Eurasia, or sheltered in the Ice Age refuges of southern Europe, before somehow, by a route we still don't understand, making their way into the Americas and becoming part of the fabric of a great native nation. If you carry R1b on your paternal line, you may be a direct descendant of a European trader from the 1700s, yes. But you may also be the descendant of a far older, more mysterious father, whose ancestry originated in the lands between the Black and Caspian Seas. This genetic revelation is not a dilution of identity. It is an expansion. It makes the Cherokee story not a regional tale, but a chapter in the history of humanity on Earth. For the established academic narrative, the implications are even more disruptive. The Clovis first model, which held that the first Americans were the big game hunting Clovis culture, arriving through an ice-free corridor 13,000 years ago, has been under assault for decades from sites like Monteverde in Chile, which are thousands of years older. The Cherokee genetic data adds fuel to a new model, the multiple migration model, or the Beringian standstill model. In this view, the ancestors of Native Americans were not a single band that sprinted across the land bridge. They were a diverse population, already containing multiple distinct genetic lineages that lived in isolation on the Beringian plain for thousands of years. This population absorbed small waves of migrants from different parts of Eurasia over a long period. Some of those migrants carried ancient West Eurasian lineages. When the climate warmed and the ice sheets melted, this mixed population surged into the Americas, carrying not just a single Siberian signature, but a mosaic of genes from across the ancient north. Some of that diversity would have been lost through genetic drift in isolated Amazonian or Arctic groups. But in the Cherokee, a large settled, agricultural population with a long history in the southeast, that ancient diversity may have been better preserved. The haplogroup H and the anomalous Y-DNA could be the fossilized genetic memory of that Beringian melting pot, a pot that included ingredients from Western Eurasia thousands of years before the pyramids were built. Let's move into the expert and witness corroboration. The lead researcher behind much of this data, Deborah Bolnick and her colleagues, did not set out to prove a transoceanic migration. In their published work, they are models of scientific caution. They acknowledge the anomaly. They present the frequencies, and they carefully discuss the confounding factor of admixture. They do not come down with a definitive claim of pre-Columbian European contact. Instead, they call for more data. They call for ancient DNA from the southeast, which has been so elusive. They call for whole genome sequencing of modern Cherokee populations to resolve the subclades and rule out, or rule in, ancient origins. And since that paper was published, other voices have entered the conversation. Geneticists studying ancient remains from around the globe have filled in the map. We now know, from a study published in Nature. In 2018, that Native American populations do contain a faint but real genetic signal from an ancient North Eurasian population that contributed DNA to both Europeans and Native Americans. This ancient North Eurasian ghost population, represented by the 24,000-year-old Malta boy from Siberia, carried markers that would eventually become the Native American haplogroups Q and C, but also carried genetic affinities to populations that would later live in Europe. So the very existence of a deep, ancient connection between the ancestors of Native Americans and the ancestors of Europeans is now a proven fact in the human genome. The Malta boy himself did not carry haplogroup H, but his existence proves that Siberia during the Ice Age was a crossroads, where genes from West Eurasia and East Asia mingled and recombined before even entering the Americas. Could a related population have carried haplogroup H into the Beringian mix? It's possible. The elimination of other theories has left this as the most plausible surviving explanation. This is not the sensational, pseudoscientific claim of Europeans were the real Native Americans. That is a racist fantasy. The ancestors of the Cherokee are unequivocally the primary original people of the Americas. But the story of how those original people came to be, the assembly of their genetic heritage, is far richer and more complex than the old textbook drawings of a single arrow crossing from Siberia to Alaska. The Cherokee genome is a palimpsest, a document written and overwritten, and we are only now learning to read the oldest layers. So, the elimination process is complete. The Clovis, eliminated as the sole ancestral source. The ancient Beringians alone, eliminated as a pure, unadmixed population from East Asia. The Norse, the Romans, the Phoenicians, the Egyptians, all eliminated as direct sources for the West Eurasian signal. What survived the elimination is the only answer that fits the known facts while remaining consistent with the broader sweep of human genetics. The Cherokee carry within them the signature of a deep, pre-Columbian connection to the populations of ancient Western Eurasia, an echo of the complex dance of migrations that occurred in the far north during the last ice age. A small founder group, already carrying haplogroup H from an ancient intermingling somewhere in Siberia or the Beringian plain, made the journey into the Americas. Their descendants mostly lost that lineage through the random chances of genetic drift. But in the Cherokee, perhaps because of the size and stability of their ancestral population in the southeast, perhaps because of chance, the lineage survived, passed down from mother to daughter through hundreds of generations in the forests and mountains of what we now call the American South. Then, thousands of years later, European men arrived from across the Atlantic, carrying a different, more recent branch of that same ancient story, and added their own lines to the mix. The result is a modern genetic profile that confounded the simple tests, that sparked controversy, and that ultimately forces us to abandon the image of a pristine, isolated America that waited in stasis for Columbus. The Americas were, from the very beginning, at the very end of the last ice age, a new world built by people who were already a mixture of ancient lineages from across the northern hemisphere. And now, you've seen the elimination. You've watched every false origin crumble. And what's left is the truth that was almost buried forever, a truth that now belongs to you. Because this isn't just a story about genetics or ancient migration. This is your origin. If this video resonated with you, if it challenged what you thought you knew, then don't let this truth stop here. Hit that like button to push this revelation further into the world, where others with Cherokee ancestry or a passion for real history can find it. Subscribe now, because the next investigation is already underway. More ancient DNA is being sequenced. More secrets are unraveling. And you will not want to miss what comes next when the genetic evidence finally speaks from the bones themselves. Drop a comment below with your thoughts, your own family stories, the myths passed down to you. The old story is dead. The real story has just begun. Thank you for watching. See you in the next one. Thanks for staying until the very end. Goodbye for now.